In the wild, every rustle of leaves, every call in the distance tells a story. A story of survival, of adaptation, and balance. But what happens when these animals live behind walls under human care? From the towering giraffe to the mighty elephant, every species tells us something unique. Understanding their well-being is not just about watching from afar. It's about listening deeply through behavior, physiology, and even the hidden world of microbes. It begins with a simple scan. Scan the QR code and opens into a new way of seeing animal welfare. This Flipbook introduces an integrative welfare assessment model that combines behavioral observation, fecal glucocorticoid metabolites (FGM), and 16S metagenomic profiling of gut microbiota. Each indicator captures a different dimension. Behavior tells us what animals do and how they cope by observing their behavioral patterns. Interpreting changes in behaviors helps us to understand emotional states and coping strategies, especially in captive environment. FGM reflects physiological stress responses. Non-invasive fecal sampling lets us track glucocorticoid metabolites over time. We examine trends and contexts, changes across weeks, and responses to stresses, including anthropogenic activities. This supports evidence-based decisions while minimizing disturbance to the animals. Gut microbiota is a diverse community of bacteria in the gut. Microbiota profiles shift under chronic stress, poor diet, diseases, or captivity changes. It can detect changes before behavioral or hormonal signals. 16S rRNA metagenomic analysis is used to identify gut microbiota taxa, understand its composition, diversity, and changes, adding a window into animals' diet, health, stress response, immune balance, and environment. Together with behavior and FGM, they form a complete picture. In our tiger case study, we observed tigers at NWRC who were born into captivity and tigers at MTCC who were relocated from NWRC to MTCC. We observed their behavioral patterns and collected their fecal samples for FGM and 16S rRNA metagenomic analysis. At NWRC, we found that the younger male tigers exhibited higher pacing, while the tigers exhibited less activity and increased resting. Females exhibited high grooming. At MTCC, tigers exhibited increased pacing, active and grooming behaviors, but decreased resting during adaptation period. For the FGM levels, female tigers at NWRC exhibited an increased FGM compared to males. Tigers at MTCC exhibited a significant decline of FGM levels across periods as the tigers adapted to the captive environment. For the gut microbiota at NWRC, females exhibited increase in diversity. At MTCC, there was an increasing trend in diversity across periods. Our findings show that tigers adapting to captivity exhibited more natural behaviors, reduced FGM levels, and stabilized gut microbiota profiles over time. These translated measures can be used for other wildlife species too. Integrating behavioral observation, FGM analysis, and gut microbiota profiling provides a more holistic and sensitive approach to assessing wildlife welfare. This triad offers insights into animals' emotional state, physiological stress, and internal health. This Flipbook bridges science and practice. By integrating behavior, hormones, and microbiota, we move from simply keeping animals alive to ensuring they can thrive.